can move on to the next talk. Now we are going to have James Taylor, CEO and co-founder of Precision NanoSystems in Vancouver, talk about meeting the challenges of microfluidics, enabling nanomedicine development from basic research to the clinic.
Okay. Great. I think I just need... Okay, excellent. Well, thanks a lot for inviting me here to talk. And here we'll be talking about microfluidics, manufacturing of nanomedicines. So just very quickly about our company, we've developed technology, the microfluidic basis for manufacturing nanomedicines. And we work with groups, really have developed technologies for the improved use and development and manufacture of nanomedicines, working from the smallest scales, really the tens of microliters, through to helping groups develop their preclinical drugs through our various instruments, and then bringing those towards the clinic with our clinical testing and product manufacturing, our Scaleup platform, which we'll talk about today. We're headquartered in Vancouver, Canada. We come from a lipid nanoparticle group, but we also do lots of polymers these days, but really set up to solve the key challenges we had in making nanomedicines. So nanomedicines, as we know, it's a fantastic field to be in. What we're seeing in the industry, it's impacting most areas of pharmaceuticals. There's a wide variety of different types of nanomedicines. Shown here are liposomes, the lipid nanoparticle systems, and various polymer systems, but really starting to be very pervasive across pharmaceutical, both the big and small companies, so very exciting times. Our company, we think a lot about how these nanomedicines are made as a drug product, so how they self-assemble. Shown here is a cartoon of a lipid nanoparticle for nucleic acid delivery. And this drug may be, say, 75 nanometers in diameter, but comprises 100,000 or so molecules that need to self-assemble to create that final structure. And how it self-assembles has a big impact in how it's made, big impact in how it works, where it goes in the body, and how efficacious of a drug it is. Our contribution to the field really has been the industrialization of the microfluidics approach for making nanomedicines. And so with this, we're taking advantage of fluidics at very small scales, principles like laminar fluid flow for exact reproducibility, and we combine that with continuous flow micromixers that allow for very rapid and controlled mixing. The reaction volumes are in the order of tens of nanoliters, but at the same time, we're able to make and produce the nanomedicines in a continuous flow environment that operates between 10 and 20 milliliters per minute. And we scale up the process through parallelization. We'll talk about that today. But also importantly, it's a low energy input system. So we often are working with macromolecules like 20 kb plasmid fragments or large 15 kb RNA fragments. And the typical protocol is you have your lipids or polymers dissolved in a solvent, and you're rapidly mixing that with a miscible anti-solvent. So say lipids dissolved in an ethanol, rapidly mixing with an aqueous, and you're forcing a self-assembly process to happen through that and nanoprecipitation, and we control that self-assembly. So with that, I just want to show why this is important. This is slides that were put together from one of our users, so this is a third-party data, but really showing the progression from top-down extrusion, shear mixing type approaches through to bottom-up self-assembly approaches, first through macroscopic mixing approaches using turbulence and the like, and then through the inline microfluidic mixing. So taking in this experiment, what they did is they took the exact same components into the microfluidic-based system, and the exact same components and buffers and everything into larger scale macroscopic mixing. And what you're seeing is the process makes a big impact. You're getting essentially different particles come out. So with the green plot here shown in the sizing data, showing a more monodispersed population with the microfluidics as compared to the inline macroscopic mixing. With that, not only are you seeing changes in physical characteristics, but also in how these drugs work. So this was a lipid nanoparticle system loaded with an siRNA against a liver secreted protein called factor VII. Through that, again, the green bar showing the microfluidic mixing approach, you get a more potent gene knockdown dosage as measured versus the blue bar showing the second generation inline macroscopic mixing. So very important. The process is the product, we heavily believe, and you can see that impact across a wide variety of different systems. And I would highlight the platform is very interesting for a wide variety of different types of nanoparticle systems. We've been fortunate to have lots of folks using our technology and have well over 60 peer-reviewed publications using this for things like polymer, liposomes, lipid nanoparticles, emulsions, and the like. So really anything that can be self-assembled. And with that, I just want to highlight a few slides around what we can do first off at controlling how we can control the characteristics of the nanomedicine, and then I'll show how we can scale that up. So this data will show liposomes and polymer data, so a typical liposome formulation, as well as a PEG PLGA polymer nanoparticle. And with this, we can use different process parameters to fine-tune things like particle size. These plots show that total flow rate, so how quickly we flow through the mixers, which is a proxy for mixing speed in these types of mixers. And using high flow rates for the liposomes, for instance, we can really drive down the particle size, in this case down around 50 nanometers. We've been able to make 20 nanometer liposomes as well. And we're seeing similar trends with the polymer system as we drive to really asymptotic mixing speed. Additionally, we can control things like the ratios between the two inputs, so the amount of solvent and antisolvent. With the liposomes, you get very well size control where we really can tune the size of your liposome on the orders of nanometers, allowing us to create a particle that is specific to the needs, be it trying to get to a different area of the body, deeper tissue penetration, or what have you. And similarly with the polymer system, however, it seems like this flow rate ratio has a bigger impact in lipid nanoparticle systems than it does in polymer systems, which probably has something to do with how these systems are self-assembling, but we don't quite know the mechanism of that. And then also just highlighting here, very robust to concentrations. So with the liposome system pushing up towards 75 megs per ml without significant particle changes, and also with the polymer system showing something similar in this plot, showing up to 60 megs per ml with that. Also, what we've been able to see is through the improved mixing, we're able to get higher drug loading, and we have various data on this for polymer systems and for liposome systems. I'll just show this plot on polymer systems. Where compared to a process like beaker precipitation, we get much higher drug loading and also better able to control particle size following encapsulation of the drug itself. And that has a lot to do with just how the mixing process is working. And then finally, I think it's really important, but maybe less sexy result, is that it's a very reproducible platform. So using this, you can really trust your data and be more rational in the experiments you have. This was done by 4 essentially non-expert operators, and really the computer control of instrumentation coupled with microfluidics allows for extreme reproducibility. Now, in the last part of the talk, I just want to talk about scale-up. So a big part of what we want to enable is people to bring these drugs towards the clinic. Want to develop a platform where you can rapidly develop things on the bench scale, but then know that you have a path forward to scale up. And through the microfluidic approach, what we're doing is using parallelization. So really doing more of the same to get to larger volume throughputs. On a single mixer scale, like I said, we're doing 10 to 20 milliliters per minute. So if you put 10 of those in parallel, you're getting up to hundreds of mils per minute flow rates. So the Scaleup system, we've developed a GMP platform where it's really designed for GMP manufacturing. It's a continuous flow pumping system with a fully disposable fluid path. And the microfluidic mixers are identical to those used in the benchtop setting. So with that, you know that what you're doing at the small scale is exact to what you're doing at the large scale, and the processes can be transferred directly up to that larger scale. And so using our typical 8 times parallelized system here, which is for smaller, earlier clinical runs, we can manufacture 25 liters in a 4-hour time period. So really fitting into the typical manufacturing runs. Data showing here with liposomes, in this case, showing that the different technology or different equipment using the same microfluidic technology can make essentially exactly the same particle. So this is done using one of our benchtop system for a 10 mil formulation, the Blaze instrument for 100 mil formulation, and a 8 times parallelization, just making a 1 liter test formulation, again, getting exactly the same particles. And this is the schematic for the clinical scale manufacturing. So essentially what we do is we continuously flow pump fluidically bus to your parallelization scheme. That then is captured on the output. Following, often we see our users wanting to do an inline dilution, so that can be done. You do have the ability to waste through transient pressurizations. Then you have your final product for processing. With this, we were also able to demonstrate significant volume throughput, a single microfluidic mixer. So this is doing a 5-liter run of up to approximately 7 grams of sRNA, and at a very high concentration, so at a 1.39 mg per ml sRNA batch. And again, you're getting essentially the same particles from the small scale. That was a 2 milliliter run to the 5-liter run. And the final product means that we've done tangential filtration following making the nanoparticles off the chips. And this was done using a lipid nanoparticle system for sRNA delivery. And then also we can see that concentrations, again, at these large scales are not having a big impact on particle parameters. And lastly, comparable to using those particles and looking at a gene knockdown study, this is, again, using that factor 7 sRNA, you can see essentially the same dosage pattern across the different scales, which is very exciting. So that's all for my talk. Lastly, I just want to highlight that right before the CRS, we put on a Nanomedicine conference that you may be interested in, some flyers at our booth, but if you're able to make it to Cambridge, to Boston on July 15, that would be great. If not, it's also live streamed, but we have some great speakers and really love to put this together. Great. Thank you.
So we can take 2 questions. Maybe I'll give a start. A very practical question. So how would you clean, or what are the dose disposable parts of such a GMP scale manufacturing unit?
It's a great question. So all the fluidic path is all essentially plastic. So the chips are injection molded plastics that we make, plus the tubing and so on. Depending on the pumping system I showed there, we'd also be either washing or throwing out the pump heads. Depending on the cost of your drug, that makes a lot of sense, depending if you want to do the washing protocols or the disposability. Mm-hmm.
Okay, thank you.
Great.
Other questions? So has anything, a system like this, gone to market yet?
No, it's a good question. So we launched the GMP system just earlier last year. So we're gearing up towards that. But no, people have not gone all the way through clinic with particles made on our platform.
Yeah, the particle size control seems to be excellent.
Yeah.
So this structure that you showed for the self-assembling nanoparticle, was it proven by X-ray, for instance, or could let's say, the number of particles aggregated in one?
Yeah, no, it's a great question. So we've done, to understand the structure of those lipid nanoparticle nucleic acid-based systems, a lot of work has been put into electron micrograph, but also a lot of computer modeling. So they have sort of nano domains within the particle and how those systems come together. And the cartoon shows even though it's happening at the same time, there is likely a sequence to how the system is all coming together.
Okay, thanks a lot.
Yeah.
Professor Scaleup?
Yeah.
Did you ever make cell handling microfluidics?
Make which ones?
Cell handling cells.
Sense?
Sorting cells, for instance.
So the systems that we use are simply continuous flow systems, so we haven't done sorting.
Bubbles?
No, we haven't. So our systems, it's a mixing process, not what typically is made, say, in droplet, when people make droplets and bubbles and so on. So we do, self-assembled nanoprecipitation reactions, necessarily microdroplet, which is another type of physics. Yeah, good question. Great.
Any more questions? You have not? Thanks a lot.
Thank you.
And we move on to